place to start. And in going back to the literature, in 1974, van der Meulen did some early TEM work to look at the aboral end in my species of coral, and here's what he found. So this is a cross-section, a longitudinal section of a larva, and on the left-hand side is the aboral end, and on the right-hand side is the oral end. And focusing on what's in the red box here, he found there are several different cell types represented in that aboral end. And so this big white one here is a nematocys, a stinging cell, and right next to it are what he calls 4 flagellated sensory cells. And I have one of them boxed in the red here. And so based on the cell morphology where he saw this basal rootlet cup and a single flagellum that extends outside of the cell body, outside the larva even, he predicted that these could be responsible in detecting settlement cues. However, this hypothesis was never tested, so this is just purely based on their cell morphology that he saw. So I thought it would be interesting, well, what if there's some way to manipulate these cells and remove them? Would larvae be able to still sense the cue and therefore undergo settlement and metamorphosis? However, these sensory cells are interspersed among other cell types, making their specific removal difficult. And in an aboral section like this one here, the actin filaments are labeled with Floydin in red, and the DAPI in blue is labeling the cell nuclei. So essentially those sensory cells that van der Meulen saw are in this region somewhere. So what if I were to remove that aboral region altogether? Would larvae, first of all, survive? And second, would they be able to detect the settlement cues when exposed to them? And so I devised this experimental design to do just that, to separate the aboral and the oral ends of larvae to see how each one individually would react to settlement cues. And on the left-hand side is a picture of an actual larva of Postdoctoral Researcher, and I did the same thing with Montipora, so I did it with both species of coral. And on the right-hand side is a schematic diagram, which I'll be using for the rest of my talk. And I've made 3 different types of cuts that separated the aboral and oral ends along the length of the larva. And so the first type of cut yielded 1/4 aboral and 3/4 oral. And the second type of cut essentially just split the larva in half, half aboral and half oral. And the third type of cut yielded 3/4 aboral and 1/4 oral. And how did I do this? I get asked a lot, how do I do this? The larvae are first relaxed in magnesium chloride, so that stretches the larva out, essentially. Otherwise, these larvae naturally tend to change shape from spherical to cylindrical a lot of the times. And so the magnesium chloride relaxes them, and then I would transversely section these larvae using a sterile razor blade. And then these sections were then separated and transferred to filter seawater to allow time for wound healing. And they were placed in the filter seawater for 24 hours. And so here is a picture of a larva that has just been cut in half. And this is the oral pole here, and this is the aboral pole. And you can tell by a thicker layer of epidermis on the aboral pole. So then these sections were exposed to several different types of settlement cues for 24 hours. I've included a natural cue, and so for Montipora capitata, that would be crustose coralline algae, and for Postdoctoral Researcher, a natural biofilm. I also included an artificial inducer, cesium chloride, at a 5 millimolar concentration. So cesium ions act by depolarizing cell membranes, inducing excitable response in cells, and they are a common artificial inducer to induce settlement in many other types of larvae as well. And the point of including an artificial control here, an artificial inducer, is to see whether settlement and metamorphosis would even be compromised when I make these cuts. So even if these different sectioned larvae are not able to sense natural cues, would they still be able to undergo metamorphosis because there might be some sort of internal signaling that goes on? And that wouldn't be surprising if we think back to that network of cells that we saw earlier. The controls included sectioned larvae in just filter seawater alone, no Q, whole larvae in the natural Q, whole larvae in the artificial Q, and whole larvae in a filter seawater control. And 5 dishes or 5 replicates were set up for each treatment with 10 sectioned larvae per replicate. And the assay was then scored by determining the percentage of larvae or the percentage of sections that underwent metamorphosis after 24 hours. So here are the results for Montipora capitata. Along the x-axis is the section of larvae that gets induced and that remains after the cut, shaded in gray. And along the y-axis is the percentage of larvae, or the percentage of sections that completed metamorphosis after 24 hours. So here are my whole larvae, and here are my aboral sections, and then the oral sections. And focusing on just the crustose coralline algae Q here, shaded in black, the black bars here, we see that it induces whole larvae, as we expect it to, a high percentage of them. And among the aboral ends here, they induced a lot of the aboral ends to also metamorphose, with increasing percentage as you have a larger section to work with. It did not induce any of the oral sections to metamorphose. So this species did exactly what we predicted, in that the aboral ends were able to metamorphose in response to the natural cue because they can sense it, and the oral ends are not able to because they can't sense it. Now looking at cesium chloride now, the artificial inducer, we see that that induces whole larvae to metamorphose. It induced a lot of the aboral ends to metamorphose as well, and it also induced some of the oral ends too. So this suggests that while the oral ends are not able to sense the cue, that hasn't taken their ability away to undergo metamorphosis. So that species did exactly what we predicted. Now now there's a different way to interpret these results as well. If we go back to this schematic diagram here, the different types of cuts I've made can be looked at this way, in 4 different parts. So quarters of the larvae. And if we go back to the graph here and look at what each of our sections contain, this section is just the part 1, so it just has that first quarter. This type of section has parts 1 and 2, so that's half of the larva, and this type of section has parts 1, 2, and 3, so that's 3/4 of the larva. And what's common among these 3 different sections is that they all have a part 1. So this suggests that part 1, that aboral tip, is essential for this species of coral, in that it uses the potential sensory cells are localized in this area, and it's necessary for them to detect the cue and necessary for them to metamorphose. Now turning our attention